Okay, lovely. So we're very delighted to have Professor Bob Shapley with us today in our seminar. Professor Shapley is a Natalie Clues Spencer Professor at the Sciences in the Center for Neurosciences at NYU, a professor for neurosciences, psychology, and biology at NYU, and also an associate member of the Current Institute at NYU. He did his MA in chemistry and physics at Harvard College. He then continued to a PhD in biophysics at Rockefeller University, where he studied neurophysiology and biophysics under the supervision of H.K. Hartline, the Nobel Prize laureate for medicine and physiology, a year after Bob arrived in his lab. His PhD investigated the firing rate patterns of the cells in the eye of the horseshoe crab. He then continued to a postdoc at Northwestern University at the lab of Christina Enroth-Kugel on cat retinal ganglion cells, and then continued to a second part of his postdoc at Cambridge University in the labs of Fergus Campbell and John Robson to study human edge detection with David Tolhurst. He then continued to study retinas of various species at Rockefeller as assistant professor, and then later on moved to NYU Center for Neurosciences, where he studies the primary visual cortex and visual perception. He published hundreds of papers, many of them in top leading journals as Nature, Science, and others, has more than 23,000 citations, and there is so much more to say about him, but I'll make my introduction, I'll shorten that. And he investigates vision from multiple perspectives. And today we're delighted to hear about your work, Bob. So welcome to our seminar.
Well, thank you very much for your kind introduction. I feel a little bit like Marley's ghost with the chain carrying it around with me. And I wanted to say, to remind everybody that I'd be glad if you would interrupt if you have a question that you'd like to get clarification about. So it's okay with me if you interject, and I'll try to ask for questions as we go along, just so we can get for clarity. Now let me go and do the logistical operation here of sharing the screen. Okay. I left it in the middle. Okay, good. So this is what we're going to talk about. I thought it would be fun to talk about color. And it's one of the things that's been on my mind for a long time. And I wanted to emphasize for this group, particularly color perception, but also make the link with the neurons that are underneath the perception and supporting it. And I'll get right to it. This is the summary of the talk. Whoops, I always have this problem, sorry. If you look in the primary visual cortex of animals, and we generalize to humans, you can divide all of the cells that respond to color into two groups, the single and double opponent cells. And we'll talk about them in details in case they're not familiar to you, but for many of you, they will be familiar. These two groups of cells combine the cone signals in different ways, and we'll go over that. The single opponent cells contribute to color detection and to threshold vision. But most of what we call color vision, what people, non-vision scientists would call their perception of color, is superthreshold color appearance. And that depends almost entirely on the activity in the double opponent cells, and I'll try to make that case for you. And then the other point that I think is more general, that might be useful for other areas of vision science and other areas of perception, is that vision at threshold is different from superthreshold perception. And it has different qualities and different characteristics, and we propose it's because of the differential weighting of the single and double opponent cells, which changes with color contrast. And we can, it's a very simple explanation that the responses of the different types of cells grow with contrast in different ways, and therefore they change places, threshold and superthreshold. And this principle or result or conclusion may have wide generality, which is to say threshold psychophysics may be only of partial heuristic importance in understanding perception superthreshold. Okay. And the other thing I wanted to say is that this work is collaborative. It's a result of many different collaborations, which I'll talk about in detail as we go along. The neurophysiological work mainly was done with, that I'm going to talk about, was done mainly with Mike Hawken and Elizabeth Johnson at NYU. And then we did some collaborative pooling of resources with Barry Lee more recently. Then I'm going to talk about neurophysiological population work that was done in collaboration with Shea Zweig and Hamutal at Bar Ilan. So bringing the coals to Newcastle by talking about the color responses in the monkey brain to Bar Ilan University. And then finally, I'm going to talk about visual perception stuff with work that I've done very recently with Valerie Nunez and Jim Gordon, my longtime collaborator on color vision. Okay, and we'll talk about them as we go along. Right. So first I want to start off with human perception and your perception. So I'll show things to you and you can see for yourselves and how much your color perception is controlled by what's going on at the edges between colored objects and their surroundings. And this may be familiar to you, but on the other hand, there's some points that you might not have thought about. So here's a demonstration. And this is, in fact, inspired by the work of the artist Josef Albers, who did these hundreds of paintings called the Homage to the Square. So my imitation of Albers is with these little squares, these 4 squares, the little ones. So they're all physically identical on your screens wherever you are. But they have different surroundings. This one's surrounded by the gray neutral background. This one is surrounded by a kind of tan version. This one is surrounded by green, and this one is surrounded by the orange. Now you may not believe that they're the same, so let me do this. So if you just put a ring around them, I use white, could use gray or black. They are all actually physically the same. And, but if I take the white away, then you see the color returns, the differential color returns. In fact, the one that's most interesting to me is this one over here on top of the background of a color that's similar to it, because it seems to be desaturated or decolorized. I hope that's the effect that you see. On a gray background, it's sort of a nice vivid yellow, but over here, it kind of looks like a pale ghost of that color. And it's physically identical. You see the same kind of thing in different colors. I just chose this as an example. Here's another one. So these are, on the gray, it looks like a sort of a bluish green or green. But on this color background, so this square is the same color as the background here, the same physical color. It loses all its greenness and looks kind of like a dull gray. The same exact object on the yellow background sort of looks blue, and on the blue background sort of looks yellow. And you can see that here. If I ring them, they're all physically the same, but sorry, I'm getting ahead of myself. But I wanted to go back and forth here. So if you go back and forth, and this, again, what's happening here is that if you surround this one with a color which is the same hue, but a different value, so it's slightly lighter, you see that the object itself loses its color and becomes desaturated, almost colorless. This effect has been known for hundreds of years. And in fact, John Mullen has a nice paper where he talks about the history of color contrast and ascribes one of the first demonstrations of this to the French mathematician Monge in like 1790 or in that era. So it's kind of from the time of the French Revolution, this effect was known. But it's interesting. And we can explain it. We can actually, we have a neural explanation for why this happens, but it's of greatest importance. Now, these effects are usually thought of as being somehow related to color induction. But what I'll show is that in fact, color, and I'll show you later that perception of color in general is governed by the same mechanisms that are causing the changes around the boundaries of these different test squares. And another way of seeing this is looking at this kind of color perception demonstration. Sorry, this one. So this is actually very much related to the work of the Gestalt psychologist David Katz, who described how color perception was divided into two groups or two classes, surface color and aperture color, or surface color and film color. And he thought of it as being part of the properties of the stimulus that caused it. And of course, it is related to the properties of the stimulus that causes it, but it's also related to the mechanisms of how we see color generally. So here, so I have to explain what I'm doing here. This is a square of color. And if I rip it out of its surroundings and just put it against the gray background, it looks like this. But embedded in all of the surroundings that it's in, it looks like this. What's happening here is that this square, I took something like this one over here, I put it here, and then I blended it with the background uniformly. So there's like a gradient of mixture between the gray and the pink. And so this square is surrounded by another square right around it, which is a slight blend of this color with the gray. And then that's surrounded by another one that's blended with the gray. And so it blends into the gray uniformly. And I hope you see it as I do, that the square just on the gray background is a lot more vividly pink or red than embedded in its surroundings. Here, this looks sort of like what Katz would call a film color. Here, this is what we would generally look at. We would characterize this kind of appearance as a surface color. And my feeling now is that the complete explanation of the difference between film colors and surface colors is having to do with what's going on at the boundary between the object and its surroundings. Here, there's no real boundary, and so it sort of blends into the background. And what Katz didn't say, and what I think is worth noting, is that if you take the same object and you make it a film color versus you make it a surface color, the quality of its appearance changes. And that's what actually attracted a lot of the Gestalt psychologists, that this looks different from this. But what's interesting to me today, and I want to make you interested in it, is that the color changes. That is to say, it's a lot less colorful when it's a film color than when it's a surface color. So the amount of color saturation is diminished if there's no sharp boundary between the object and its surroundings. And what I wanted to show in these previous demonstrations, like in this one, is that here, these are two surface colors. You're not going from surface to film, and yet you can diminish the saturation again by making the boundary not so different in color. So if you make the colors similar at the boundaries, you can diminish the apparent color. And the question is, how do you explain that? And so that's what my story is, to explain that. And just to show you that this is not true only for red and green, there's also, this is the blue version. So this is a stimulus which would mainly stimulate the S cones. And if you rip it out of its surroundings, so you take it from being a film color to being a surface color, again, it's a lot more saturated as a surface color than it is as a film color. And all of these, so what I'm trying to argue here is that ordinary color perception of surface colors depends very much on the boundary. And the boundary is responsible not only for the qualitative appearance of it being a surface, but also that it's colorful. The color is conferred on it by the boundary. Okay, so then, one thing I wanted to say, sorry, I forgot, is that in looking at and thinking about cats and Gestalt psychology, I remembered a very nice quotation in a book called Organization in Vision. I don't know if you can see it, by Kanitsa. This is a famous book. I don't know if you know about it. It's published in 1979. And let me just read the quotation, which is related to this Gestalt exploration of color. He says, so he did other demonstrations that are sort of like what I've just been showing you. And he says, the above observations indicate that chromatic qualities should not be considered merely as the material on the basis of which perceptual space is organized. Instead, one must also take into account the effect of spatial organization on the perception of the chromatic aspects. So that's a fancy way of saying, or maybe it's the translator's way of putting his Italian, that it isn't only that color affects space, but space affects color. And therefore, he concludes in this quote that I've used before, that is, space and color are not distinct elements, but rather are interdependent aspects of a unitary process of perceptual organization. So another burden of my talk, a subtext as it were, is that color and form are not dissociable into separate segregated categories, but actually they interact. And this has been many people have pointed this out before. I'm just trying to harp on it here. The color is not like painted on. It's actually emerging from the spatial pattern that of elements that you're looking at. So the question is then, how do we do this? So I'm going to look at some neurons, aspects of neural activity in the retina as seen through the lateral geniculate nucleus, and then neurons in the primary visual cortex, the striate cortex. And we're going to study their visual properties as how they might be related to these visual perceptual aspects of color depending on form. And I use two tricks to study color responsive neurons in monkeys. And I'm going to be looking at the spatial chromatic properties. So these two tricks are, one, the classic trick of studying visual spatial neurons with grading patterns. This is the famous Robson demonstration reproduced by Izumi Ozawa, where you study the spatial properties of neurons by looking at the response to patterns which are periodic in space. This is a swept grading, so it's not periodic, but if you look at any small patch of it, it looks quasi-periodic. And then the trick is to use the spatial frequency tuning properties of neurons to infer what the spatial properties are. So that's what I'm going to talk about. That we did a lot of experiments where we used grading patterns because they were convenient to use, because for various technical reasons, they're easier to use than bars or spots. But from the pattern of activity, the tuning curve that I'm tracing out, you can infer the spatial tuning curve of the spatial profile of the sensitivity of the neuron in space. And so, for instance, particularly, I should point out, if you have a low pass function that I'm tracing out, like this, which is a Gaussian function of spatial frequency, that corresponds to what you'd find for the spatial frequency tuning curve for a Gaussian function of space. That is to say, one which has no negative component. And this more coarsely tuned one with the coarse, this tuning curve of this mechanism indicated by the coarse dashes is traces out the spatial frequency tuning curve of this coarse dashed profile down here. So that we can, if we see a spatial tuning curve studied with a series of grading patterns that looks low pass, we can make the inference that the spatial tuning curve is monophasic. And if we see a spatial tuning curve that's tuned, where there's an optimum and it falls off on either side, we can make the reasonable inference that it's a tuning curve which has sort of a Mexican hat kind of organization. So that's one trick. That's one trick, is the spatial filtering allows you to determine spatial structure. Question?
Yeah, yes, Bob, I wanted to ask, so if you are intending to study also surfaces, would that correlate or correspond to the very low spatial frequencies? I mean, how would you study a surface? Would you consider that the very low spatial frequencies?
Well, it's interesting that you say that, because people use that term surface to describe the neurons that are responding to low spatial frequency. In fact, people make the connection, surface neurons to low spatial frequency. But in fact, the curious thing is that surface color is actually being responded to by neurons which are spatially tuned. I mean, that have a kind of a more complicated profile.
I see.
It is, let me put it this way. If you have, people have used the label surface to describe low-pass neurons. So that's the simple answer. The answer is yes. That's okay. But the more complex answer, I want to leave you to keep in the back of your mind, is that, in fact, when we see surface color, we're not using surface neurons. We're using edge neurons. And so we'll see that later.
Okay.
So part of the, I mean, a summary of my whole talk is that, in fact, we see surfaces through edges. Even though there are surface neurons, they're not the ones that actually respond to the surfaces. But that may sound crazy, but you'll see it. It makes some kind of sense. So the one trick is using spatial filtering to study the neurons. And the other trick is to exploit the fact that single cone receptors are colorblind. So it means you can choose two very different colored lights that are equally effective in exciting a cone. And one cone cannot tell the difference between these two cone metamers. So if you modulate between two lights that are equivalent for a cone, the cone doesn't respond, and so it's silent. And so you can look at the other ones that are not silent. So another way of putting this is that you can make a silent substitution pair, which constitutes a cone isolating stimulus, because it isolates the responses of the other cone that's not silenced. And the reason for doing this is that, as you know from basic physiology in the retina, the cone photoreceptors are characterized by their spectral absorption curves. This is, sensitivity versus wavelength from Baylor, Nunn, and Schnapf. And if you just use two monochromatic lights that are at equal heights along the curve, and you modulate between them, say for the L cone, modulate between, I don't know, 670 and 520 or something, the L cone won't respond to it, because it's not a change for the L cone. The absorption is the same. And therefore, modulating between those two lights would be silent for the L cone. But the M cone, here indicated in green, whose response is much less at 670 than it is at 520, would be modulated a lot if you go between 670 and 520. So it's not silent for the M cone, and therefore the M cone can be isolated. You can use, so we use this trick to isolate cone inputs to different neurons and sort of gather how they work by looking at their cone inputs. So a lot of the work that we did is actually cone photoreceptor inspired. We're trying to understand how the cones work and how they're wired to the different color neurons. And we got a lot of mileage out of understanding that. I should just say, these are the same curves plotted, linear versus wavelength or log versus wavelength. So it's just to show you how these things look on different plots. So we use these two tricks, and we create stimuli which are, say, L cone isolating or M cone isolating. We can also make them, make modulation between colors which are equally luminant. So they're equal for the luminance mechanism, but not for any one of the cones. And then we can use luminance stimuli. So these are the kind of stimuli that we use to study visual neurons that are interested in color. And I should say, you might notice that the L cone isolating stimulus looks quite different from equiluminant. The M cone isolating stimulus doesn't look so different from equiluminant, and that's just the way it is, because the L cones contribute a lot to luminance. But And these are not exactly what we use. This is just kind of a heuristic to show you the idea that you can generate different kinds of stimuli using different colors and different balances. So we used these kinds of stimuli to study visual neurons, both in the LGN and in the cortex. And this is work we published in a paper with Barry Lee, work that, this is the paper that we published. This is work that Mike Hawken and I did together. And this shows two different cells. The luminance responses are indicated with the black. So these are the response to luminance gratings. The red is the response to isoluminant color gratings, red-green color gratings. And then these are the responses, the orange, the yellowish, brownish, and the green are the L cone and M cone isolating responses. So this is the response to L, and this is the response to M. And the main thing two things. One is that the L and M responses generally were low pass. This is two different neurons, but they're in the whole population, they were very low pass. And then the trick that I said before, using the spatial filtering to deduce what the cone inputs look like in space, we derived these spatial profiles. This is for one neuron, this is for another neuron. This one, it was L plus M minus. Here, this was M plus L minus. And they were, just Gaussian-like in space. The cone inputs had a kind of Gaussian blob spatial profile. And therefore, now this is somewhat controversial, I have to say, but at least there's consistency between several labs and inconsistency with one lab in Seattle about what this looks like. But generally speaking, most of the data of the in vivo visual system is consistent with the idea that up to the level from the retina to the LGN, the color cells are responding with cone inputs that look like this, which means that the spatial profiles look like this, which means that the bottom line is that a caricature, a cartoon version of what the input is to the cortex from the eye to the cortex is that it's made up of a population of single opponent cells. So we call them single opponent, where the L cone, the M cone has one polarity and the M cone has another, and the L cone has another polarity, and they're spatially overlapping, and one may be slightly bigger than the other, although there's a lot of spatial overlap in fact, and they have opposite signs, and the only opponency, the only sign difference is between the cones. But within a cone type, there's just, there's net input of one sign. So this is the caricature of the input to the cortex, the single opponent cells. And as I pointed out, their characteristic feature is that the cone inputs are low pass. Also, if you look at the responses to pure color stimuli, that's the red curves here, they're also low pass. And that's what you predict from this, you could predict this, that these should be, that the response to isoluminant color, pure chromatic stimuli, should be low pass also. And that's what the input, the spatial input is to the cortex. It's this low pass color input. And then when you get to the cortex, you can look at this, you can use the same tricks, the same two tricks, spatial filtering and cone isolation, to look at neurons in the monkey cortex, which we use as a model for the human. And if we take a look at a cross-section through the monkey cortex and a primary visual cortex, we see it's a layered structure. The input layers, the output layers, layers 2 and 3 are the output layers that go to the rest of the cortex. And a lot of our work has been studying the neurons in the outer layers because they are ones that are projecting to the rest of the cortex, and they're the basis for color perception. Okay. And if you look at neurons in the cortex, you find that there is a population of single opponent cells. And the way you can tell that is that if you look at the response to equiluminant color, they are low pass. They don't show any downswing at low spatial frequency. And if you look at the cone inputs to these single opponent cells, they sort of look qualitatively like what you see in the LGN. They sort of look low pass. And the other thing about them is that almost all of these single opponent cells, well, not almost all, a large fraction of them are getting only L versus M input and very little S input. And then there's some of them that are a very small fraction that are getting S input. And these neurons respond very much more strongly to color than they do to black and white. So over the region of spatial patterns that they will respond to, the color response is much bigger than the response to luminance. And for this reason, they've attracted a lot of the attention of people who study color in the brain. They've looked at these neurons and they said, oh, these are the ones I want to study because they respond so much better to color than to luminance. They must be the ones that are important for color perception. But in fact, these are a minority of the cells that respond to color in the cortex. And I'll try to make the case to you that they may contribute to color perception, but they're not so important as these other cells, which are not so low pass. Just want to complete the loop of the reasoning I talked about with the two tricks business. Going from the spatial organization, going from the spatial organization, we can go to the, going from the spatial frequency filtering, we can go to the spatial organization here. So we can also map these stimuli, these cells with spatially. And we've done this some, and we get these kind of spatial maps. This is now a two-dimensional spatial map, not a profile, of the L cone input to one of these single opponent cells and the M cone input to the single opponent cell. And you can see that it's monophasic, and so the color indicates the sign, positive and negative. It's all positive for L cones and it's all negative for M cones. Sort of looks a little bit like a big LGN cell receptor field. This was done, stuff that was published in a paper with Elizabeth Johnson and Mike Hawken a while ago. And so you can go from the spatial frequency map to the spatial map and measure it directly and verify that the two are consistent, not just infer it. But it turns out that these single opponent cells, as attractive as they are to color scientists, are a minority of the color cells in the cortex, and that most of the color cells in the cortex look different. And if you look at their spatial organization, well, let's look at their spatial frequency organization first. These are the so-called double opponent cells, also exhibited in this paper. So this is again along the spatial frequency axis. They show spatial tuning for both color, that's the red, and for luminance patterns, and very similar spatial tuning. That's a characteristic of them also. Lisa Thorell and Russ Develoy had seen these cells first many years ago. And then also Peter Lenny and John Krauskopf and Gary Sklar studied them also. We emphasized how important these were for color, I think that, and for color perception. And by looking at the cone inputs, we could sort of see that the cone inputs were tuned the way the pure color stimuli were tuned. So these were different from the LGN input. They had a different spatial organization for for cone isolating stimuli and for the response to pure color stimuli. So they were spatially tuned for color. And if for this neuron and for neurons like it that we could map, we could see that the spatial organization was different from the single opponent type. Each cone type had both positive and negative regions, and that each, where there was a negative region for the L cone, that is the L cone response would be, the cell would respond with spikes to a decrement of the L cone input. It would respond to an increment of the M cone input. And here it would respond to an increment of the L cone input. And at this location indicated, it would respond to a decrement of the M cone input. So that locally the cone inputs were opponent, but they were opponent in one sense in one location and in the other sense in the other location. That is to say, it was L on, M off at this location, and it was L off, M on at this location. So there was double opponency. And this neuron was in fact tuned for color. But it also did respond to luminance. It is so that these.
Bob, can I ask a question?
Yeah, sure. Of course, you can always ask a question, please.
Were they responding directionally selective?
These neurons are not direction selective. No, the direction selective cells are segregated in the magno pathway. So if we can go back, I'll show you. Wait a second. That's a very good question. Thank you for asking. This is the other talk that I didn't give, which I'm working on, is the direction selective pathway. And if we look at the layers of the cortex, the magno input layer is 4C alpha. The parvo input layer is 4C beta. These cells that we're talking about, the single and double opponent cells, are mainly in layers 2 and 3 driven from 4C beta. They're in the parvo pathway. The magno pathway is where you see the direction selective cells in 4C alpha and in 4B. These are, magno input comes into 4C alpha, then it's relayed up to 4B, and then goes out to MT into V2 and so on. And the direction,
I was asking because of the displacement of the receptive fields, it might suggest that moving in one direction will activate in one way and moving in another direction.
That isn't what causes direction selectivity. Okay. So so I'm giving you too much information, more than you wanted to know. But in fact, the direction selective cells are segregated into 4C alpha and 4B. We're talking about a different parallel pathway, as it were. Yes. So the other thing about, but since you mentioned direction, I'll just point out that these color double opponent cells are also orientation selective. They can respond to different orientations with selectivity. So with the equiluminant versus the black-white stimuli, they can be orientation selective in the same orientations. And these are the most numerous color cells in the cortex. And we think that they are most important for color perception. Because of the kind of demonstrations that I showed you, the single opponent cells wouldn't be particularly important for responding at color edges, but a neuron like this would be very sensitive to a color boundary and would respond very strongly if the color boundary moved one way or the other. So our cartoon of the receptive fields of these neurons are that they have cone inputs which are opposing in different locations, and that they're sort of organized in this odd symmetric way. So these are like color edge detectors. And there's other work that I haven't, that I'm...
excuse me. Does it have to be edge rather than bar selective, odd rather than even symmetry?
It turns out that, as I show, they turn out to be like this. So, I mean, most of them turn out to be odd symmetric, or I should say, tending towards odd symmetry. They're not all exactly odd symmetric, but they're, and another thing to point out is that there's other work that I'm not talking about by other people. This is a largely densely populated area where people have looked, and there's some nice work that was done by Conchetta Marone around the time we were doing this, indicating that many color responsive neurons in human are in fact odd symmetric. And that's, I quote them in some of my review articles, you can find it there. Okay, so so we think that this characterizes most of the neurons. And in fact, I would say odd symmetry is more the rule than the exception among visual cortex neurons, not just in the color system. And if we look at the spatial frequency tuning, going back to this spatial frequency domain, and look at the, just a second. Look at these double opponent cells and compare them to single opponent cells. This is a paper done by Dennis Schlippick and Steve Engel using the data from Elizabeth Johnson and Mike Hawken and me, but doing the work that we didn't do of actually looking at the population average spatial frequency tuning for non-opponent cells, that's a solid curve, double opponent cells, that's the dashed dot curve, and single opponent cells, that's the dashed curve. And the single opponent cells, they're low pass, and they tend to cut off at fairly low frequency. And the double opponent cells actually have pretty much very similar spatial tuning to the non-opponent cells that are not color selective at all. And this is a feature that we exploit later in the human work, that they respond to different spatial frequency domains, as it were. Now I want to talk about the work that Zweig and Hamatal did, studying this problem using voltage sensitive dye imaging. And I'll go through it very fast, but it's a summary. It's sort of, other people had looked at how these double opponent cells could be sensitive to edges, particularly, I should say, van der Heijt's group in a very nice paper by Friedman, Zhou, and van der Heijt. But here we were able to look at the whole population simultaneously by looking at voltage sensitive dye imaging. And what was done was to present colored squares on a gray background. These are equiluminant colors, monkeys fixating, and this is presented in a place which will show up in the window where they're measuring the voltage sensitive dye fluorescence. And these are pictures of the monkey's brain, looking at black squares in the top row and red squares in the second row. This shows the time course of the response to red and black over the whole region of the window. And then you can see not the whole square, but the sort of the top, the left-hand edge of the square. What's image is not the part near the fovea, but this part over here. The response in the cortex to this part of the square is imaged in the window. And you can see it emerges, and then you can sort of see the outlines of the square here. And this is the outlines of the square with black. And it emerges over time, over the time course of the stimulus. The stimulus is shown for 300 milliseconds, indicated by the black bar. And you can see that there's a heightened response where it's red, and where it's white is a very heightened response. And where it's greenish and bluish there's less response. That's a lower response. This is the color scale of how much the fluorescence changed with time. And even out here, say halfway through the time the stimulus is on, the response is very heightened along the boundary of the red square, and it's smaller in the middle. There's a hole. And this is what you'd expect if most of the response that you're measuring is coming from double opponent cells. You would see a response near the boundary, and the double opponent cells in the middle, there'd be no boundary, so they would be stimulated less, and so the response would be weaker. And another way of looking at that is to look at a profile through the square as a function of time, and that's indicated here. This is for black over here, and this is for red. I want you to focus on the red, and it just shows you the response of the center of the square and the response of the cortex that's located near the projection of the edge. And here's the response. And this profile, this position along here is position along this slice through the image, the voltage sensitive dye image. So this is the center of the square. These are the two edges. And this is time progressing. And what you see is that there's a big response on the edges. This is one edge, and this is the other edge. This is one edge, and this is the other edge. And in the center, there's a minimal response all the way during the time that the stimulus is on. That is to say, there's a hole. And that's what you'd expect from if the response is dominated by double opponent cells, and they're most of the color responsive cells in the cortex. So what they're showing, and what they showed here, is that in fact, this is the image of a uniform square, a surface in the cortex is in V1 anyway, it's dominated by the response to the edges. And it's all the time that it's on, it's dominated by the response to the edges, meaning that in fact, the picture that the brain has of the external world is a caricature filtered through the spatial filters, in the case of color, of the double opponent cells. And in general, it's what the brain is getting is a caricature of the outside world. which needs to be reconstructed. That is to say, we don't have an isomorphic picture of the world that we can refer to. There's not some internal image that the homunculus can look at, but in fact, what's encoded is patterns of filtered versions of the world which needed to be interpreted. And so perception is not at all just driven from the outside, but in fact you need to take what the neural response is and do some kind of thinking about it in order to know what you're looking at. So I think this just writes down what I was saying. That is to say, the hole in the middle of the response to red is very important, and it's a result of the double opponent filtering of the outside world. So I'm just writing here what I just said, so I don't have to go over it. And I would say color is a model that we understand fairly well of everything. That is to say, all these things that I'm saying about color also generalize. That is to say, what the brain is getting is not a picture. It's getting some caricature which needs to be interpreted. So the last part, I just want to have a few minutes to talk about the perceptual results. They're more recent, but in a sense, this is what puts some perceptual meat on the claim that I make that the double opponent cells are most important for color. Because based on the ideas of before now, you might have thought because color is painted on and it's low pass and it's surfacey, that the brain would integrate color signals over large areas, and thus you think that large colored objects would be more colorful. But if you believe double opponent cells are producing the signals for color, then you'd expect that fine color patterns might be better. Remember, going back to this, going back to this, if in fact the double opponent cells are most important for color perception, you might expect that fine patterns that activate the double opponent cells would be most colorful. And if you thought that the single opponent cells were most important for color, you'd think that low spatial frequency, coarse patterns, blurry patterns would be most important for color. And it turns out, I wouldn't be talking to you if I thought that it was the blurry patterns. But I should say, the background for why people thought that large blurry patterns would be most colorful is based on this classical work by Kathy Mullen about what is important at threshold. At threshold, humans are most sensitive at low spatial frequencies. So if you just measure a human's detection performance, it looks low pass. And so from what I talked to you before about the tricks of studying color vision, you'd think that the neurons that are responsive for color would be low pass, it would be Gaussian blobs of some kind, and that blobby colors would be most effective in producing color perception. But that's not correct. So what we did using, this is the work with Valerie Nunez and Jim Gordon, we used a method derived from Hervich and Jameson of scaling. We asked people to scale how much color saturation they saw in an image. And we asked them, if the gray background has 0 and pure color has 100%, what percentage would you give to the color that you're looking at? So this is pure scaling psychophysics. And the stimulus subtended about 2 degrees. They had a half second to look at it, and then it faded out, and then it was equiluminant with the background, and then they rated how colorful it was on a scale from 0 to 100. And we compared blurry squares with blurred edges with checkerboard patterns, which had the same space averaged color. So if you average the checkerboard over space, the actual cone catch would be the same here as here. But here it's gathered into a fairly fine spatial frequency checkerboard. And the question is, if we compare these kind of patterns, which looks more colorful? And the answer is very clear that the checkerboard is much more colorful. So people, they're equated for space averaged, the space averaged cone activation is the horizontal axis. And for a given space average cone activation, the apparent saturation of the checkerboards is much greater than the square. This is for stimuli which are red. That is, we use stimuli which look like this. They were these colors. It was red. It was a red-gray checkerboard versus a red square. And then we also looked in cardinal directions of color space, because to look at different color mechanisms, because the red activates all cones, L, M, and S cones in different ways. So what we wanted to do is just activate the LM axis and the S axis separately for color aficionados. And the answer is that in the LM direction also, that the checkerboard is much more colorful than the square for the same space averaged cone contrast. And in the S direction, so if we isolate just the S cone, we have the same result. It's not as big a difference, but it's definitely there. So bottom line is that for all color perception, in fact, what we're using is a mechanism which is spatially tuned and which is not responding best to the lowest spatial frequencies, but is in fact tuned for pattern. And just as a demonstration of the results for your own eyes, these two are equated for space averaged cone contrast, but these look a lot more colorful than that. I mean, it's a very robust result. I mean, it's not controversial or anything. You can get it replicable again and again. No one departs from it that we've tested in the lab. But it's a little unexpected because people tend to think of color as being this low pass, blurry, painted on system. In fact, it's a spatially tuned system that is important for, and it makes some sense why there are so many cones, because you need to have spatial resolution for the color system. Okay, so I have a summary of the visual results, which is that, as I showed you, single and double opponent cells combine cone signals in different ways. And superthreshold color appearance, as I just showed you, depends on activity in color tuned cells, which we think are the double opponent cells in V1. Therefore, at a mechanistic level, color and pattern are linked inextricably in human color perception. That is to say, it's not because of, I mean, it may be that the neurons are like this because of external influences which have developed over evolution, but the mechanistic reason why they're linked inextricably in our heads is because they're going through the same neurons. And single opponent cells may be important for color detection. They are important for color detection, and they may be used for aperture colors, for film colors, for color atmosphere. But the double opponent cells seem to be telling us about color appearance of color surfaces that we ordinarily see, like, you know, Knitz's book, or a red tie, or, you know, the painting on the wall. So the summary for color vision is that we have a special pathway that we're using a lot for color perception. But I want to say something else, that I have a second summary, which is more general. And that is that threshold properties, and the properties I'm thinking of are like spatial summation or spatial tuning, don't apply to the superthreshold visual system in the realm of color anyway, because colors, superthreshold properties depend on edges and pattern and so on. And in the case of color, it's easy to explain. You have two different mechanisms and their responses have different contrast dependencies. So of course, superthreshold, one mechanism can take over, and near threshold, the other mechanism can be more sensitive, and they switch positions depending on how well they grow with contrast. It's not hard to understand. But it may be true that this is a principle that applies not just in color perception, but to all perception, that superthreshold is a different perceptual world. And therefore, we should work, and I think it's a good idea to develop techniques which can be used to measure what's going on superthreshold, not just at threshold when we're studying perception. Okay, that is it for me, and I'll stop sharing. Okay,
so I'd like to invite everyone to unmute yourselves, and let's give a applause to Bob for a wonderful and inspiring talk. I have some questions of my own, but I'm welcoming others to open their microphone and ask.
Yes, I have a question, Bob. Great talk. I really enjoyed it.
thank you.
Wouldn't you predict then that if I look at a square, I should see the colors near the edges more saturated than in the middle of the square, and I don't think I experienced that.
So that's a very good question. And the answer is that when we're looking at the square, we're taking the information from the edge and extending it all the way in. And indeed, Van der Heide has written about this question of filling in, that the filling in is actually being done at a high level, not at the low level. So the low level is providing the input, but what we see is not what is at the low level. I think this is congenial to you.
Exactly. So basically, you saw a nice demonstration that V1 is not involved in color perception. It's just setting up the input to the color perception.
Well, it depends how you look at it. I mean, what I would say is that it provides the input to color perception. So, I mean,
perfectly agree. Just like the retina.
Well, but it does more than the retina does because it's doing more work. But indeed, it is providing. Let's put it this way: if you take V1 away, you don't see the red square. And you can't be veridical. You have to check what V1 tells you to know what is veridical. But it is what is being given is not an isomorphic picture of the world. And in fact, I would say the way to understand V1's role is that it is sort of like the external world for the cortex.
Perfectly agreed. Yes.
So I think that let me just say that this view and the work that Shade Zweig did with Hamutal and on this whole in color perception, I think is very important. I think it actually tells you, it changes your idea about how the external world is represented, not just for color. Color is just a nice example of it, and it points. And this is very similar to Van der Heid's view, I should say, about color. That you're not looking at an isomorphic picture. You're looking at a rendition or a filtered version that needs to be interpreted.
Can I just add to that? Can I just add to that, Bob? Nice talk, but are you really talking about looking at the color of objects versus looking at the color of backgrounds? Because the input that you need is the color of objects or faces or something like that. And the color that you don't need in such accuracy, if you like, and perceptually, you don't need the background. And so if you had more spatial items in a particular scene, then your color perception is enhanced by something like attention or something. Second question, what's
wait a second, wait, let's do one at a time. Hold on.
Okay. Well, it might be related to my second question. What is V4 doing? What is V4 doing in all of this?
Yeah, so so the first question is that you can get these effects not particularly with attention. I mean, you can, in fact, you very often, the biggest color edge effects are in peripheral vision when you're attending somewhere else. And so I don't think attention is the issue. However, it is true that figure ground effects have a role to play in how vivid the color is. So there definitely is there are higher level effects than simply edge contrast that play a role. And this goes all the way back to the Gestalt psychologists who showed very nice examples of when something is figure, it becomes more colorful, and when it's ground, it's less. And I actually showed you an example of that in my talk.
Yes, yes.
Where I had a little square which was the same as the background, and the color of it when it was a background was very different from the color of it as a square. So it is absolutely true that color perception depends upon whether something is figure or ground. But in a sense, what I'm trying to say is that the mechanism for making it figure is the double opponent cells. That is to say, they are the ones that give you the information that there's a figure here. And the single opponent cells are actually responding mainly to the background. They respond to large areas. So they'll respond to the background the same way they'll respond to a figure. Now, the second question about V4, it's very interesting. And I neglected to say something. So you give me a chance, an opportunity to say something that I meant to say. And that is that Many of the neurons in V4 have very similar properties to these cells in the upper layers of V1. And it's also true, as Carl Gegenfertner and Daniel Kuiper and their colleagues showed much earlier, in V2 and in V3, there are many color neurons which are color tuned and also spatially tuned. In V4, there's a whole wide range of different color types. There's a very nice paper by Bushnell, Pasupathy, and Baer about neurons, a population of neurons in V4 who are especially active just for equiluminant color and for nothing else, sort of like the color preferring cells in V1. But in fact, the great majority of cells in V4 are like the great majority of cells in V1 and the great majority of cells in V2 that respond to color in being spatially tuned and responding to both luminance and color, like the double opponent cells that I showed you. So V4, I think, is a very, it's a higher level of color processing. It's closer to the color templates in the infratemporal cortex. But its color properties are not strikingly different from the upper layers of V1.
Okay, thanks.
Can I ask something? Hi.
Yeah, sure. Who? I can't see.
I'm Yossi Mandele. Thank you very much. It's a great talk. We found in the past that anomalous trichomats had better spatial vision, higher contrast sensitivity and visual acuity.
Then whom? Which?
Anomalous trichomats, colorblind, anomalous trichomats.
Oh, anomalous, right? Yes. Mm-hmm.
And do you think that this could be something to go up to the cortex, or could it be some adaptation in the retina? Can you think about any potential?
So this has been studied a lot, and I've actually speculated about this, of course, that color has a cost. That is, if you have good color vision, you lose something. You lose spatial vision as part of the cost. My feeling is that it's a property of the whole pathway. That is to say, it's not just, it's a retinal property and it's also a cortical property, both, because you could have, how to put it? I mean, the thing is that if you're anomalous trichromat or if you're a dichromat, you don't have as much diversity in your cones. That is to say, they're more alike each other. And therefore, optically, you're in a better position because you don't suffer so much from chromatic aberration. You can focus on the one set of wavelengths that's in focus for your one set of cones. And therefore, everything is better for space. You're no longer, so it might be the whole system is optimized in such people. It's a small effect, but I think it's an important effect, and it might be, worth tracing. But one of the problems with color vision is that you have these cones. I showed you the cone spectral action spectrum. They're not identical. I mean, they overlap a lot. One of the reasons that they overlap so much is probably because of spatial vision. If the cones were too far apart, the chromatic aberration would be so bad, your spatial vision would be terrible. So you have to. So there's a compromise. You have to have the cones separated, but you don't want to separate them too much, or else you won't be able to see sharply. So when you see, the dichromats or the anomalous trichromats, they lose color vision, but they gain the fact that they don't have chromatic aberration anymore. That's my feeling.
Yeah. Interesting. Thank you.
Oh,
yeah.
Great talk. But how does this story square with the fact that color TV signals could get away with much less bandwidth in the color domain than they can in the luminance? And we're not working at threshold when watching TV.
Yeah, but TV doesn't look right. That's why TV looks artificial. I mean, the thing is that we have a lot of spatial vision for color, and it is true that they get away with it. That is to say, you can get away with it. You can say, oh well, it's the same. It's red. the uniform is red or something. But if you actually say, can I discriminate this from reality? The answer is yes.
So are you saying to provide really convincing color television, we shouldn't be wasting our bandwidth on high definition? We should be using it in the color part of the signal.
Well, it depends what you want. You know, it's like I just said with responding to Yossi Mandel about the anomalous trichromats. It's a trade-off. The primates had to, you know, they went for color vision, but they had to make a trade-off because they also wanted spatial vision. So you have to divide your bandwidth, you know, however you want. And, you know, there's only so much bandwidth, you know, that's the ultimate limitation in life, is how much bandwidth you have. So the answer is you could get better color vision if you devoted more bandwidth to color, and it would look more real for color, but then you'd be giving up some resolution about exactly, you know, what's the boundary location and so on. So, you know, it's all bandwidth, you know, I mean, that's all there is.
Thanks.
Shaul?
So Bob, thank you for this talk. Can you get a Craig O'Brien corn sweet illusion?
Yeah, so that's a very interesting question. And in fact, Hamutal has been after me about this for years already. And the answer is that you can, but it's not as good as with black and white. So I've emphasized how similar the double opponent cells are to non-opponent cells, and how they respond, they contribute to color vision in the same way that the achromatic cells contribute to spatial vision in achromatic vision. But they're not identical. And in fact, the single opponent mechanism is stronger for color than it is for black and white. There isn't such a strong non-opponent black and white signal. And so the way the Craig O'Brien corn sweet illusion works is that you take advantage of the fact that you can't really see very well at low spatial frequencies. So you can create an illusion where there's this missing fundamental. You leave out the low stuff and you can't tell. In color, that wouldn't work because you detect the fact that that there's something missing. But you do get very strong edge effects. So you can create patterns where you grade the edge down and it looks like it's filling in, but it's never quite as satisfactory because you detect the deviation from uniformity at a fairly low level. But you can create such things, okay. And there are and there are definitely, as I showed you, edge enhancements. But
right.
But you can't use the same exact
all the way. Yeah. Okay. So then, do you want to say something about color assimilation?
It's another whole topic. I mean,
yeah. Okay.
I mean,
okay.
But yes. It's another whole topic. I mean, assimilation one thing I would say is that it's a more high level thing than this. That is to say, it's cortico-cortical interactions, and it hasn't been investigated as much, and we haven't investigated it as much. But indeed, it's a very important effect, but it's not something that I have good data to talk about.
You think it's not V1?
No, I think it could be partly V1, yes, because of edge-to-edge interactions. So, I mean, there's definitely, recurrent interactions between neurons will cause assimilative effects. But so I think that they are there, but but by higher level, I mean, it involves more of the cortical interactions within the network, which we haven't studied. I mean, this is sort of like a first pass through the network before we look at the interactions, I would say.
Peter, do you want to ask a question?
Yeah, sure. Bob, can you see me?
Yes, I can.
Thanks for the talk. I just wanted to follow up on the last point you made, so about developing techniques for comparing spatial tuning.
Good, excellent.
Threshold. So I'm not going to get into the details of psychophysics. I mean, there are techniques like that in psychophysics, but they're being used very rarely. And as far as I know, only in the context of luminance signals. But so if you want, we can get into that. The bottom line there is that what you normally find is that at threshold, in fact, spatial tuning is sharper than a super threshold. It's a surprising counterintuitive result. So that's a whole different topic within psychophysics, and one can get into the question of whether those techniques address the same phenomena you would see, for example, in the absence of noise, which is vital to some of these approaches. But the question I have for you is, in neurophysiology, I would have thought that's much less of a problem because you can just measure response in spike rates, so you don't have to worry about this issue that in psychophysics, in order to measure the tuning, you need to do the threshold, and so you've got this situation where you need to manipulate contrast to get the threshold itself that gives you the tuning, and so as you know, in neurophysiology, when you record from neurons with luminance signals, what you find is the opposite of what I just said. A low contrast, you get low pass, and a high contrast, you get high pass. Now, what is the current take in the...
No, wait a second. Hold on. What result are you referring to there?
So the literature that I'm aware of in neurophysiology, recording typically from V1, is that a low contrast, you get low pass tuning in neuronal responses, and a high contrast, you get high pass or band pass.
That's not what we've seen. I have to say.
With luminance signals.
Yeah, with luminance signals, the orientation tuning or spatial frequency tuning doesn't, if anything, it kind of broadens as you go to high contrast because it saturates.
Okay, that I would be interested in because the literature I'm aware of goes in the opposite direction.
Well, what literature are you talking about? I'm not following you.
Let me look it up because it's...
So why don't you look it up? Because I actually, my own experience is that contrast is very interesting. So people talk about contrast invariance, in fact, of tuning curves like spatial frequency and orientation. In fact, it is true. So, I mean, it doesn't go from low pass to high pass, but if you go from low contrast to high contrast,
yes,
the orientation selectivity can be slightly higher at high contrast. That's right. So,
well,
spatial frequency, but spatial frequency doesn't change all that much. But orientation changes a little bit. It gets a little more tuned. That's true. That's correct. Sorry, I wasn't following you. So yes.
So you would agree on, yeah.
Yes.
And my question is whether that also applies to color selective neurons like double opponent cells, or if it's only a property of luminance signals.
So actually, I don't know that people have looked with color contrast
exactly
at the selectivity of the neurons. I think that hasn't been done, and I would expect, like you, that the neurons might become somewhat more selective at high contrast just because of network interactions. Yes, I think that is possible. That's an interesting idea.
Well, I mean, the thing is, it's actually the opposite in human psychophysics. So in human psychophysics, what you normally find, surprisingly, is that at low contrast, you get sharper tuning, more bandpass tuning.
Well, this is for achromatic.
Yeah, for luminance signals. Well,
yes.
The thing is, I'm not aware of any study that has looked at this for color signals, for isoluminance signals. I don't know of any study that has looked at this. So, but for the luminance signals, you get something that goes in the opposite direction of what you get in the electrophysiological literature. And I don't know about color vision. That's why I was asking you. And I thought maybe in neurons, somebody had looked at this problem, but it doesn't look like there is any literature on this.
No, I don't think so. I mean, the thing is that, how can I put it? We can piece together the situation in color is different from luminance. As I mentioned in a way to Shal's question about the Craig-O'Brien-Cornsweet illusion, in that you have two mechanisms that are not so unbalanced. That is, say, the double opponent mechanism is more powerful at high contrast, or has a bigger response, and the single opponent mechanism has a higher response at low contrast. In achromatic vision, that isn't really true. It's the tuned mechanism is stronger across all contrasts. So the relative weights of the color and luminance are not the same in that respect. But so what happens with color is that as you increase color contrast, cone contrast, you're switching roles. You know, the single opponent one is dominant at low cone contrast, and the double opponent becomes dominant at high color contrast. And that switch doesn't happen. I think that's what, so the results might be different for color and luminance in this respect.
Okay, thank you.
But Bob, is there time for more questions?
Sure. It's early in the morning for me.
Okay, I've got a question about following Jan's question about the V4. I want to ask about the globes or the high order visual areas, and you can say IT, or the areas, Doris Tsau's studies and Bevel Conway, color sensitive areas, more downstream that have been found to be more closely related to the, let's say, perceptual space, or let's say color space or color circle or whatever. So I want to ask, how do you think these inputs are then combined? I mean, these are the inputs that flow into those areas, or you think that they are also combining additional signals, and where's the transformation? Because, okay, that's my question more or less. How do you think this is getting?
Well, the thing is, my own understanding is that the work by the people you mentioned and by others who are there, who worked on the area also, like the Japanese, I'm blanking on his name, but there's a big Japanese group that's done this. I mean, we don't really, not as much is known about the cone inputs and the way that they're combined and the, how space and color are handled. The only study that, that I cited in connection with V4 in response to Jan's question right at the beginning was that there's a lot of similarity. In fact, I mean, if you look at them the same way, they look similar.
So it's.
I think that let me just say that I think that what I glean from the infratemporal cortex literature is that color and form are highly intertwined. And that's exactly what I would expect from what we see at the early level. That is to say, neurons are selective for, they may prefer some colors, but they also prefer color not in the abstract, but also
attached to form.
Attached to form. And this is my theme song, which is the color and form are not to be separated. So so I read it in a very biased way. I mean, I'm looking for a particular angle, but
okay, thank you.
That color should be segregated in the sense of that color should be the feature that is segregated in one region versus another region. Makes sense also from a functional point of view.
Thank you.
Okay.
Yeah.
Okay, everybody satisfied?
Yeah.
Bob.
Great.
Sorry, another question. You never mentioned blob, the cytochrome oxidase blobs. Are both your single opponent and double opponent mechanisms similarly laid out in V1, or is there a difference there?
Yeah, so so the first answer is I don't really know. The second answer is that I don't know that it matters. I think that from all that we have studied, and I would say the blob, non-blob distinction has been, hasn't really carried a lot of weight, I would say. So I think that I view this as a population of color responsive neurons, and they're responding to color and form together. The people who have, I mean, we couldn't find any segregation of, we did look with fairly careful techniques at at cytochrome oxidase blobs and tried to find some functional segregation, and I really could find very little. The other thing about it also is that the actual modulation of cytochrome oxidase staining is not that large, and can be enhanced with image processing techniques, but is there's a lot of cytochrome oxidase all through layer 2, 3. So I'm not a big fan of cytochrome oxidase blobs. Another thing about it is that you may know about this study that I was a very minor partner of with Tony Mopshin's people studying the owl monkey. The owl monkey is an animal, a nocturnal monkey that has no S cones and has one L cone and has no color vision whatsoever. Jerry Jacobs talks about it in his evolution papers as an animal which clearly evolved to have no color vision whatsoever. It's nocturnal. It has big eyes for a nocturnal animal and so on, and has the most beautiful cytochrome oxidase blobs. I mean, it's this beautiful, the most beautiful system, and also squirrel monkeys that are like the archetype of the blob system that Livingston and Hubel studied. All the males are dichromats. So, it isn't so clear that it's important to understand the importance of blobs for color vision. I think that they may be important for spatial vision, but I don't see that I don't see a lot of segregation. Let's put it that way.
There are people who...
There's a question from Ruti.
Yes. I want to ask about the color and form, that you say that they go together, but behaviorally, we know that people can selectively attend to color and ignore form and vice versa, and they cannot do it, for example, for hue and saturation. So how do you reconcile the behavioral data with your claim that they go together?
Well, yes, I really don't know about the attention literature that much, I have to confess. I'd like to know, how well you can prove that you're really attending to the color. What can I say? I can't really speak to this question. It's conceivable that they're using this appeal to higher levels in the brain where color may be segregated into different qualities that you could attend to different qualities of color. I mean, clearly, we have a sensation of red versus green. I mean, they're not, even though they're different objects and different spatial qualities. So indeed, we give the same labels to things which are associated, but so why are they associated? And the question is, where is that done? I don't know the answer.
Let me ask the question in a different way. I think there are people who are colorblind cortically, not because of something in the retina. Does this on the opposite happen? Can they be form blind but still see color?
Yeah, DF. Sorry to interrupt.
Okay. No, very good, Sharon. Okay, so
I don't know this patient. Tell me about DF.
DF is the famous patient from Goodale and Milner, where she's not that, she's visual agnosic, acquired visual agnosia, but and her lesion is mainly in areas corresponding anatomically to LOC, but she is okay with doing actions, more or less. There's many papers about her. And I remember, if I remember correctly, she's able to recognize a banana by the color, but not by the form. So if she sees a blue banana, she won't be able to say it's blue. It's a banana, but she'll be able to say it's blue. So the yellowish, kind of like but maybe I'll look it up and I'll send you some. I mean, I don't know if it's relevant, but.
I'd be interested because, I mean, to my way of thinking, there can't be pure color. It has to be somehow related to form. But the question is, what? And so I'd be interested to find out about that. Thanks.
Yeah, so I'll look it up where I can find this this aspect of where it was reported. I do recall that. Okay, so I guess
what I would say is, let me just say, one of the things that I want to get across is that it's possible to have color without form. That is to say, using the single opponent mechanism, aperture color, in my way of thinking, is color without form. It's sort of like formless color, but it's crummy, it's meager, it's colorless compared to the vivid colors that you can get from the double opponent form system. So maybe I can retire to a safer position where I'm not saying that all 100% of color is surface color, because indeed I never claimed that 100% of color was surface color, but all color that's not aperture color, all the vivid stuff that makes us what we think of as normal color vision, not aperture color, not film color. That color is, I would say, is related to form. And it's conceivable that there may be some other formless aspects of color which are going through this this other pathway. Meager and crummy though it is. Okay, thanks very much, everybody.
Thank you very much. I'd like also again to thank you for a great talk. Pleasure that you joined us, very inspiring. And next week we have Ella Striam-Amit. She'll be talking about visual cortex organization and individual differences in blindness at the same time. She's also from the East Coast. So I'm welcoming everybody to join. I'll send you all the information. So thanks for joining us. And thanks again for the wonderful talk.
Leatroad.
Bye.
Leatroad, Bob. Bye.
Thank you. Thank you.
Bye-bye.
Good to see you.
Okay.
Bye-bye. Yeah, nice to see you guys.